Good evening, my name is Lindsey McCoy, and today I'm going to be presenting my research on the effect of heat shock time on the induction of lysogeny in W3104 bacteria. So before we get into the research and the study, we're going to do a little bit of an introduction and a background on a few things that are important to understand the research. So first we're going to talk about bacteriophages. So what is a bacteriophage, or a phage for short? A bacteriophage is a virus that targets and attacks bacteria specifically. They're very abundant in nature, the most abundant microorganism found in the environment. So why are these phages important enough to study, and why do they matter? And so the reason that these are important in nature is because of regulation. So because they target and kill bacteria, they help to regulate the levels of bacteria in the environment. So if one species of bacteria happens to become super high and is wiping out other species, bacteriophages are able to come in and kind of even out that level and make the environment more balanced. So phages can undergo two different types of life cycles. It can undergo a lytic cycle or a lysogenic cycle, and we're going to talk a little bit about these later. But a lytic cycle is when it kills the cell by lysing it, and then lysogenic is when, instead of causing cell death, it kind of incorporates itself into the host cell. And so then we're also going to learn about temperate phages, which are phages that are able to show both life cycles and undergo both life cycles by switching in between. So that's what we're going to be looking at in this study. And the phage in this study is going to be transferring from lysogenic to lytic, and we're going to talk about why. So next, we need to understand what lysogeny is. So what is lysogeny? Lysogeny is basically the lysogenic life cycle option of a phage, and this is when it inserts itself into the host genome in the DNA of the host and does not cause cell death. So why is lysogeny important, and how could it be used in medicine? So why are we researching it? So an example of lysogeny in medicine would be oncolytic virus inhibition. So an oncolytic virus is a virus that targets and attacks cancer cells. So to use lysogeny in medicine, we could inhibit oncolytic viruses and help them to make them target cancer, and maybe that could be a form of combating cancer. And then lysogenic conversion is just basically a continuation of lysogeny, but it's talking specifically about when it inserts itself into the host genome, certain things that it may bring along with it to permanently alter the host. So this can alter the host phenotype and things that it shows in ways that it acts. And this can increase the virulence or the toxicity or danger of the bacteria. So for example, if a phage brings with it a toxin and it undergoes lysogeny and it inserts the toxin into the bacteria, that could make the whole thing essentially a lot more virulent or dangerous. So a few previous studies on lysogeny that I've found in studying induction of lysogeny, previously scientists have looked at slowed growth of bacteria, UV light, they've used a repressor, and even altered the medium to see how this could maybe induce lysogeny. But today, what we're going to be talking about, what I used in my research, was heat shock. So then what is heat shock? Heat shock is essentially how it sounds. It is just heating up the substance, or in this case, the phage, enough to shock it, so a high enough temperature to alter how it's going to act and create a certain action or effect. So heat shock is used in other forms in science and other previous scientific studies. An example is they've used heat shock in the transfer of bacterial DNA, so that's been done before, but today we're going to use it to induce lysogeny. And why did I think this could be a possible method of induction for this study? So this kind of goes back to the phage being temperate and switching from lysogenic to lytic. So when the phage is lysogenic, it's incorporated with the virus or the bacteria, and so it kind of lives with it. And so this creates a non-ideal environment. So when the phage gets heated, it's going to get super uncomfortable. So that's going to cause it to want to maybe jump out and go into the lytic cycle to essentially survive and save itself from the intense heat, the non-ideal environment, and continue to live and multiply. So the point of my research, kind of why it is important, so lysogeny can help, or this research can help determine how to prohibit or inhibit lysogeny of phages for specific purposes in medicine. So these could have a lot of positive or negative effects, lysogeny can. The objective is to determine the impact that the heat shock time has on the transfer of the temperate phage between two different bacterial strains. And so after looking at all these factors, I hypothesized originally that as long as the bacteria is heated longer, then there will be more plaques created from the bacteria. So now that we know all about that, how did I go about doing my research? So I did 4 trials, and there were 4 replicates per trial. So I had 3 different time periods, and then I had a control, which was not heating at all. So we heated, I heated the phage for 10 minutes, 15 minutes, and 20 minutes. I did that 4 times, so 4 different trials. So there was one slight, what you would call, like, I guess, a limitation. So I did have to use 2 different host, or like receiving strains. One of them was K12 and the other one was C600, because in between a few of my replicates, or my trials, one of the strains died off. So I ended up getting around the same results, as you can see, so it shouldn't have been too much of a limitation, but that just needed to be noted. So I used 10 minutes, 15 minutes, and 20 minutes, as well as a control. And then once I heated the phage, the bacterial strain with the phage in it for these time periods, I removed it from the heat, and then I plated it on your typical medium with top agar. And then once that hardens and is solidified in there, then you plate the receiving strain, either the K12 or the C600, on top of that with top agar. And then these plates were incubated overnight, and I observed the plaques the next day using PFUs, which is plaque forming units. So this is a graph of my results. So average PFUs versus the heat shock time. So you can see our independent variable is the time. So we have the control 10 minutes, 15 minutes, and 20 minutes. And then the dependent variable is the number of PFUs that were shown per square centimeter. So as you can see, contrary to my hypothesis, at first the number of plaques did start to rise as it was heated, but then it seems to hit sort of a peak and then decrease after 10 minutes. And so we'll talk about why that could have happened a little later. So these are the average number of PFUs over the range of my 4 trials. These were the average numbers of each one. So I had around 15, 30, and then it jumped back down to around 15, and then it even went down to less than 10. So to analyze these results, I did do 2 methods of statistical analysis. I performed an ANOVA test, and the ANOVA test produced a p-value of less than 0.05, which shows that my results are statistically significant. And then I conducted a post hoc test, and so that's what these little stars are to indicate. The post hoc test showed a 0 mean difference between the 15-minute and control values, showing that they were essentially the same, and there was not a whole lot of difference between no heat time and 15 minutes of heat time, which shows that there is some sort of something happening between maybe 10 and 15 minutes or somewhere around there, that it goes back to essentially not having been heated at all, those kind of results. And then just to show the difference between that, the post hoc test also indicated that the 10-minute and the control had a mean difference of 14.33. So there was a significant difference between the no heat time, the amount of plaques that produced, and the amount of plaques that the 10-minute heat time produced. So as we looked at the graph, it does show an increase and then a decrease in the PFUs, which is also basically the level of lysogeny, or amount of plaques in the bacteria. So as we discussed, there was an increase from no heat time to the 10-minute heat time, and so the reasoning behind this would most likely be an induction of lysogeny, which is what we were researching and what we were looking for, indicating that 10 minutes of heat time definitely makes the phage uncomfortable, and it is a non-ideal environment, so it's going to jump out and become lytic to continue to survive, basically. So it's not stuck in with that bacteria in that hot environment. But then we saw a steady decrease from the 10 minutes to the 20 minutes. It just kept continued to decline, as you can see, steadily. So I would guess that this is indicating cell death, so that they were heated so long that the cells just started to die off. And so that's why there are such less PFUs, because so much heat shock was starting to kill them. So as I mentioned before, I did do an ANOVA test, and so it compared the plaque forming units, and the post hoc test compared each time period with one another to show kind of how different the results were between the different time periods. And as I said, the p-value was less than 0.05 on the ANOVA test, which indicates that the data was statistically significant, so there was definitely reasoning behind this, and so that we did what we were intending to do. So this shows, the results show that my hypothesis was partially correct. It did start to induce lysogeny by heat shock, but then we got to a point that it started to kill off the cells. So there's probably a peak at some point that it reaches before it starts to, not work any longer. So that's what it says here. And then it starts to cause the negative effects of cell death. So this shows a trend that could actually be used in areas of medicine and other areas of science. So to use heat shock and lysogeny, as I mentioned before, to inhibit oncolytic viruses, to target cancer, that's a big one. And then transduction is another process involved with phages, and it's a form of horizontal transfer. It is when a gene is transferred between two organisms. So this can be done through between humans, and it can be used for all different types of viruses. And so this could be used for future medical research in that area. Also, going back to lysogenic conversion, it has been used in vaccines. So an example was a paper that I read in doing this research. An example is cholera. And so when they were studying that, as new strains are produced and a gene was added that had the bacteria to hold a toxin within it, the vaccines could be used with, goodness gracious, the phages that did not contain the toxin. So that could be used to combat the actual virus. And then a lambda phage is something we haven't really discussed yet, but it is a temperate phage. It is one of the most commonly studied and well-known temperate phages. It has been used super common and done a lot in the field of science and research and medicine. And so some examples of that is the lambda phage was actually credited with the first real insulin production, and then also it can be used in gene manipulation, which is basically altering how a gene acts through the transfer. And this was used with the Salmonella virus. So to conclude, my research showed that a short amount of heat shock time induces lysogeny in the W3104 bacteria strain, most likely due to the discomfort of the phage, like I said, or the non-ideal environment for phage life or growth. So heating the infected phage for too long, though, will start to kill the bacteria and prevent the transfer of the phage between bacterial strains. So it would be nice, possibly in the future, as a study to maybe test closer together times, such as like 10 minutes, 12 minutes, and 15 minutes to really capture where that peak would be of efficiency for these methods. Other studies could be to test other bacteria strains to see if this works the same through all of them or if it's just a specific type. See if long amounts of heat shock are an effective method to eliminating unwanted viruses. So as we saw, we were trying to induce lysogeny and not kill the cells, but maybe we saw heating them for so long and the cells started to die off. That could also be a good thing for viruses that we're looking to eradicate. And so we could look at it that way. And then we could also determine if the right amount of heat shock could aid in the lysogeny of oncolytic viruses. As I mentioned previously, that's one of the biggest things. And so these are my references. Thank you so much for your time.